My name is Kamalani. I'm also a research associate this year with the Pioneers, and I'm advised by Dr. Tomasz Zajkowski. And today we are just going to look at how I evaluated peptide condensation in prebiotic droplets. Okay, so I'm going to start by giving a detailed introduction to a phenomenon called liquid-liquid phase separation, which you will have may have already heard. I'm going to really briefly mention LLPS is a physical phenomenon where a solution spontaneously separates into a dilute phase and a dense droplet phase due to the interactions between molecules. So what's happening is these blue molecules here are interacting with each other more strongly than they're interacting with the green molecules, which causes them to stick together and form this cluster here in the center. So you can think of this like oil demixing in water, which I think is an example many people would be familiar with. And the result is a compartmentalized, membraneless droplet. So this phenomenon is really simple, yet very, I would say, prevalent in cellular biology and in theories surrounding the origin of life. Okay, so here we see just a few of the many biomolecular condensates that form as a result of LLPS, and I just have this here to illustrate how such a simple physical process is so prevalent in our cells and how functional these droplets really are. Okay, so we talked about oil, but fatty acids are not the only species that do this. So as Medu and Cameron and the other pioneers have briefly mentioned, there are these protein sequences or peptide sequences called prion-like domains and they end up routinely exhibiting some form of phase separation. So prion-like domains are part of a class of intrinsically disordered, low complexity protein regions that are typically rich in amino acids that we consider to be stickers. Those are these kind of nonpolar or aromatic, meaning they have this ring structure, amino acids that tend to stick together and they will promote LLPS. The other amino acids here would be considered spacers, and these are amino acids that break up the sticker interactions, which are also very important because they will mediate stickiness, they will prevent kind of out of control aggregation. So here are some common stickers and spacers, and then on the right we have the interaction between stickers and spacers driving the process of LLPS, and I'm interested broadly in the LLPS behavior arising specifically from these disordered low complexity peptide sequences. Okay, so let's see. When it came to my project, what I really started to wonder is, can this innate behavior of disordered peptide sequences, this LLPS behavior, be utilized in prebiotic contexts, meaning before the origin of life? So what I'm, wondering is these short, disordered, low complexity peptides were likely abundant on primordial Earth, and how could their phase-separated condensates that they create interact with the surrounding prebiotic environment? So I became very interested in this question here, which is, can LLPS condensates formed from these domains contribute to pre-enzymatic peptide bond formation? Okay, so let's think now about peptides. So when we are thinking about prebiotic peptides, I like to discuss what I call the water problem. So the condensation reaction, and when I say condensation, I mean water gets released between two amino acids is unfavorable in bulk water solution, meaning that the formation of peptide bonds in prebiotic aqueous environments would have been very challenging. We also know that water actually does break bonds that form, so we not only need a mechanism that supports bond formation, but we need something that can keep it stable over time as well. So there have been a couple ideas proposed around this. For example, wet-dry cycling, which is a geochemical phenomenon between alternate wet and dry cycles. This would be like tides coming in and out over rocks. Then we have mineral surfaces, which could sequester and intercalate amino acids to provide these basically small reaction sites. And what I'm most interested in are called coacervates, which are these really small aqueous microdroplets formed from LLPS behavior with bulk solution. Okay, so what is a coacervate? We've already discussed liquid-liquid phase separation, and coacervates are just a type of liquid-liquid phase separated condensate. They're these tiny bubble-looking droplets here on the left. They're often associated with protocells and origin of life theories. So like all LLPS condensates, coacervates form through phase separation from a bulk solution, and we have these dense droplets and then this bulk dilute phase on the outside. On the right here is a diagram showing the very many functions these droplets have been shown to perform. They're simple but very, very robust. They can partition reactants, they can exclude competitors, they allow free in and outflow of certain chemical species. And they protect formed products. So you can see why this is so promising as a mechanism for a sensitive process like peptide condensation, because a coacervate would solve both the problem of sequestration of the amino acids and long-term stability by shielding the peptide products from bulk water. And remember, we know that many disordered regions do this. They undergo or contribute to phase separation behavior in aqueous solution, which means that if peptides condense within these droplets, coacervation behavior could be continuously enforced, which would form a positive feedback loop. All right, so I expect the coacervate to sequester amino acids, and I also expect it to provide a shielded environment for the products, but what I don't know is if it can actually promote condensation itself. So the interior of a coacervate can be very different from the bulk solution, and that means we could see increased condensation, reduced condensation, or no change at all. One of the features of coacervates that was of primary interest to me was the dielectric environment. So the relative dielectric permittivity of a solution would represent basically the solution's polarity and its ability to reduce electrostatic forces between charged molecules. So high permittivity solvents like water would easily solvate and separate ions, where low permittivity solvents don't do this. That would be something like oil. What this figure here is illustrating on the right is the high variability of dielectric conditions within different coacervates. So water has a relative permittivity for reference of around 80, and this is denoted by the letter epsilon. And then whereas the permittivity for coacervates can drop down to less than 10, depending on how they exclude water. So the dielectric permittivity of a solvent can drastically kind of affect reactions by altering transition state energies and screening ionic charges. Okay, so that's the premise. So what I'm wondering now is how we go about determining this. So the point of my experiment is to look at the behavior of an amino acid condensing onto a peptide in a coacervate environment. So I need to choose an amino acid to act as my incoming residue, and I also need to choose a peptide chain to condense onto. So I ended up choosing phenylalanine as my amino acid because of my interest in aromatic amino acids and prebiotic phenylalanine synthesis, and also because phenylalanine is a good sticker amino acid that would promote LLPS. For my coacervate scaffold, I chose a valine aspartic acid dipeptide, seen here, based on the prebiotic relevance of valine and some interesting ionic properties of aspartic acid. So the idea is that the dipeptide would represent a local site of a much larger coacervate complex. My phenylalanine comes in and condenses onto it, and then I end up with this tripeptide being formed. So I used a program called Orca to perform my calculations. Orca is something called an ab initio quantum chemistry program, meaning it performs calculations from the bottom up. That means it uses only empirical mathematical laws and no experimental data. So I ran something called a dual level thermodynamic solvation cycle, which just means that I calculated a very precise energy level for my molecules in a gas state and then corrected for all of my solvation parameters using some faster calculations. This gave me a measure of Gibbs free energy at each dielectric condition, which I then normalized to a baseline in water. So to encapsulate the wide range of possible coacervate dielectric states, I chose to run my calculations at these dielectric conditions. So epsilon at 80, which would be akin to water, 60, 40, and 20, where 20 would represent a very hydrophobic oil-like environment. Okay, so what do we get all of this? We see that as dielectric constant decreases, meaning the environment becomes less polarizable and more oil-like, the reaction does become more and more thermodynamically favorable because delta G gets lower. So actually, as it looks like as we get lower and lower, delta G starts to decrease more and more. So in a coacervate environment, our condensation reaction would seem to be favored, and this supports the hypothesis that coacervates could have acted as sites for prebiotic peptide synthesis. So just to conclude, so our takeaway here is yes, coacervates consisting of disordered peptides could facilitate prebiotic peptide condensation. And with this result, I've developed some additional questions that I'm hoping to address in the coming year as I continue my work. One is, can prebiotic LLPS structures conserve peptides over time, or will the coacervate age and collapse into an aggregate if we are only as disordered regions get more and more complex? And secondly, could the coacervate also serve as a site for the synthesis of more complex, particularly aromatic prebiotic amino acids? And ultimately, I just want to kind of leave off with this overarching idea that these three concepts, I think, start to converge on, which is, could LLPS tendencies of these nascent disordered peptides have templated the prion-like domains that we see now in the modern day biological structures by coordinating RNA peptide interactions, possibly prior to the evolution of DNA? So amino acids get synthesized in the coacervate, condense onto those pre-existing peptide chains, form longer disordered regions, and then the coacervate grows and facilitates interactions between peptides and RNA. So these are all questions I'm hoping to continue working on. And with that, I'm happy to address any questions, comments, or concerns. And thank you guys very much for listening.